students. Welcome to Priya's Life Science Academy. This is your educator Priya. Today I will teach you a very important topic of cellular and molecular biology, which is Lac Operon. And this topic is very much important for those who are preparing for CSIR NET, GATE, ICMR, NEET exam. So I'll request you all to pay attention and listen to the lecture. So let's start our first lesson, that is Lac Operon. So before starting, we must understand that what is an operon. So a operon is a functioning unit of DNA containing a cluster of genes under a control of a single promoter. So here you can see there are gene X, gene Y and gene Z. These three genes are under the control of a single promoter region. Okay, so this is known as operon system. So a cluster of genes which are under the control of a single promoter region. So by this what we can understand that an operon contains multiple structural genes and all of which are under the control of a single promoter region. So from here you can see that a polycistronic mRNA will be produced from these genes. So what is a polycistronic mRNA? So these genes, from these genes, mRNA will be produced, and from this mRNA, lots of proteins will be produced because there is gene X, gene Y, and gene Z. So from gene X, this mRNA will produce. So this mRNA is mRNA X, this mRNA is mRNA Y, this mRNA is mRNA Z, and from each of the mRNA, each of the proteins will be produced. Protein X, protein Y, and protein Z. So a single mRNA codes for more than one protein. So this operon system is based on prokaryotic system. So only in prokaryotic organism we can see this operon system, and we can see polycistronic mRNA. So polycistronic mRNA, poly means many, cistron means gene segment. So lots of gene segments are together in case of the mRNA, and this mRNA can produce more than one protein. Okay, but in case of eukaryote, we can see the monocistronic mRNA. So what is monocistronic mRNA? That each gene, suppose gene X is under the control of an individual promoter, gene Y which will be under again an individual promoter. So in case of eukaryotes, we can see single genes are under the control of single promoter, but in case of prokaryotes, lots of genes, cluster of genes are under the control of a single promoter region. So this is mainly operon. I hope you understood what is operon. Now coming to our today's class, that is what is Lac Operon. Now you may wonder that what is Lac Operon. So this Lac codes for lactose. Okay, this Lac codes for lactose, which is a sugar molecule, and this lactose, Lac Operon, this lactose is used as a carbon source in case of E. coli. This Lac Operon is operated in case of bacteria E. coli. E. coli uses glucose for its biochemical reactions, but when there is an absence of glucose, they use lactose. Like we need energy source to do our daily activities, likewise bacteria do need their energy source to do their metabolic reactions, to perform their metabolic reactions. So the most simplest molecule which the bacteria can use to do their daily activities, that is glucose. They need glucose to perform their biochemical reactions. But what happens when glucose is not present in the medium? Then Lac Operon comes into play. Then the bacteria uses this lactose as a carbon source to do their biochemical reactions. For example, I'm giving you a very easy example. For example, I like to eat rice, but whenever there is absence of rice, I'll shift for another carbohydrate source, or will I starve? I'll not starve, otherwise I'll die. So for that, I'll shift to another carbohydrate source. I may shift to rotis, to potatoes. Okay, these bacteria also have some backup reaction which will help them to survive in the absence of glucose. That's why Lac Operon here comes into play. I hope you understood the basic thing. So why we are reading this Lac Operon, you must understand that why we are reading the chapter or why we are reading any kind of lesson. So we are reading Lac Operon because in case of absence of glucose, bacteria uses this lactose as their carbon source for their daily activities. So now first look into this Lac Operon. We have already understood the operon system. Now let's look into the specific operon, that is Lac Operon. First I'll show you the three genes, lacZ, lacY and lacA. lacZ produces Beta-Galactosidase enzyme, lacY produces permease enzyme and lacZ produces transacetylase. Okay, so these three genes are in the cluster form and they are under the single promoter region. Here is the promoter region and here is the operator region. Okay, for this promoter and operator region, there is another gene known as lacI. This lacI produces inhibitor. Okay, lac inhibitor, and then this is the promoter of the lac inhibitor. Okay, so this is the overall structure of the Lac Operon. These three genes, lacZ, lacY and lacA, are under the control of a single promoter and operator region. Okay, as I've told you that this lactose will be used instead of glucose, but in order to use lactose, it also needs to be converted into glucose. So after the entrance of lactose inside the bacterial cell, the Beta-Galactosidase enzyme, it breaks down the lactose into galactose and glucose. So these glucose can be used as their energy source. Okay, that is why Lac Operon has these three very important genes. So I have shown you that lacZ produces this Beta-Galactosidase enzyme, and this Beta-Galactosidase enzyme is important for breaking down the lactose into galactose and glucose, and that glucose can then be used for their energy source. Okay, but there is another function of Beta-Galactosidase. Beta-Galactosidase also converts lactose into allolactose. Okay, this allolactose is the main inducer molecule of the Lac Operon. Okay, so what is the inducer molecule? Yes, Lac Operon is an inducible operon. Okay, point to be noted, it must be induced. Okay, because in case of normal situation, whenever there is a presence of glucose, Lac Operon need not to turn it on. But in case of this adverse situation, whenever there is absence of glucose, Lac Operon needs to be turned on. So for that, you need Beta-Galactosidase enzyme, because Beta-Galactosidase will convert lactose into allolactose, which will be the main inducer of the Lac Operon. Eventually, I'll come to this. Now, this is a very, very important area. You have to understand that when the Lac Operon will turn on and when the Lac Operon is turned off. Okay, this is a very, very crucial thing. You need to understand this very nicely. The main thing is that Lac Operon will stay turned off whenever there is a presence of glucose, because if you have your preferred meal, you will not shift to any other kind of option. So if you, for example, if you love biryani, if you have biryani, you will not shift to plain rice and pulse. Okay, so the thing is that, so bacteria loves glucose, so whenever glucose is present, the Lac Operon will be turned off. This is a basic simple funda. That whenever you have glucose, Lac Operon will be turned off. It will stay turned off. Okay, lacZ, lacY and lacA, these are the main structural genes which produce Beta-Galactosidase, permease and transacetylase. Okay, but what happens in normal situation when there is a presence of glucose? What happens? So in this case, in normal situation, whenever there is a presence of glucose, Lac Operon is turned off. So these genes, they do not produce any kind of protein. Like lacZ can produce Beta-Galactosidase enzyme, lacY can produce permease enzyme, and lacA can produce transacetylase enzyme. Okay, whenever you know transcription, whenever RNA polymerase binds to the promoter and the RNA polymerase can transcribe the gene and can produce the mRNA, but in this case no mRNA will be produced. Why? Now the answer is hidden in this region. I have told you that before the promoter and operator region, there is another region which is known as lacI+, and which produces lac repressor. Okay, so lacI+, upon transcription, it produces the mRNA, and from the mRNA, lac repressor protein will be produced. And this lac repressor has the affinity to bind with the lac operator region. Point to be noted, these lac repressors can bind to the operator region. Okay. So whenever there is a absence of lactose, this lac repressor can easily bind with the lac operator region. And as a result, what happens? As a result, RNA polymerase can't bind with the promoter. RNA polymerase can't recognize this region. Okay, that's why RNA polymerase can't go straight and can't transcribe this region. This transcription is halted. As because the operator region is masked by this lac repressor. Okay, I'll show it to you again to give you a better clarity. So if this operator region is already blocked, this operator region is already blocked by the lac repressor. Here you can see the lac repressors, they are bound with the operator region. So, and the promoter region, you know, the promoter region are the site where the RNA polymerase enzyme can bind and can start the transcription. If you see here, you can easily see that if this is a gene, this is the promoter region, and in the promoter region, you know that RNA polymerase enzyme can bind and they can transcribe, and from the DNA, mRNA is produced. But if here at the operator region, if the operator region is blocked by a repressor, how the RNA polymerase can go and start the transcription of the entire gene? So the RNA polymerase is halted. It can't recognize the region. So as a result, the genes are not transcribed and Lac Operon is turned off. So in the absence of lactose, Lac Operon is turned off. Okay. Now we will see that at what position Lac Operon is turned on. Yes, you guessed right. Whenever there is a presence of lactose and absence of glucose, this Lac Operon will be turned on. Here you can see that even if the lacI genes are producing the repressor proteins, but the repressor proteins are masked by the allolactose, by the lactose inducer. Okay, so here from lacI, the repressor proteins are produced, but the repressor proteins are masked by this inducer molecule, and in this case our inducer molecule is lactose, but lactose is converted into allolactose, which is a potent inducer, and it is converted by Beta-Galactosidase enzyme. Okay, so this allolactose can block the repressor molecule from binding to the operator region. So the operator region is free. Now the RNA polymerase can sit and can run the transcription process, and from these genes this mRNA is produced, which contains lacZ, lacY and lacA, and once mRNA is produced, the consecutive proteins are also produced, that is Beta-Galactosidase, permease, transacetylase. Okay, so Lac Operon is turned on only in case when lactose is present and glucose is absent. I hope you understood the conditions when the Lac Operon is turned on and when it is turned off. In this case, when glucose is not present, so bacteria tend to move to another carbon source, which is lactose. So lactose enters inside the cell and it helps to turn on this Lac Operon by blocking the repressors, by blocking the inhibitor, the lac repressors. Okay. I hope you have understood the both of the condition. Here I'll write for you Lac Operon turned on in case of whenever lactose present and glucose absent, and is turned off whenever glucose is present. Yes, When glucose is present, no need to turn on the Lac Operon and lactose is absent. Okay, so this is the basic rule you must understand. I hope you have understood the basic points whenever when Lac Operon is turned on and when Lac Operon is turned off. And I hope you have also got the answer why Lac Operon is known as inducible operon. Okay, because in natural condition it is turned off. It needs an induction to turn on, and the induction is produced by lactose. Whenever lactose enters inside the E. coli cell, then the Lac Operon is turned on. Otherwise, it is turned off by the lac repressors. So, to turn on the Lac Operon, you need an inducer molecule which is lactose, and the most potent inducer is allolactose, which comes from lactose only by the conversion of Beta-Galactosidase enzyme. And after the Lac Operon is turned on, these three proteins are produced. And what is the function of these three proteins? Beta-galactosidase will convert lactose into galactose and glucose. Then the glucose can be used for their energy source. Permease helps to transfer the lactose inside the E. coli cell. I'll come to this point in the next class. And trans-acetylase transfers the acetyl group. But not much function of trans-acetylase is known so far. So this is the scenario. I'm giving you a quick revision. So this is the thing. Every organism needs their energy source to do their daily activities. So as E. coli. So E. coli needs glucose to turn on their biochemical reactions. But whenever there is absence of glucose, the E. coli have another backup method, that is to turn on the Lac Operon. In normal condition, this Lac Operon is turned off by the help of the lac repressor. And in case of and whenever there is an absence of glucose, the Lac Operon needs to turn on, and it will be turned on by the help of the lactose molecule. What will happen? The lactose molecule will block the lac repressor from binding to the operator region. So the operator region is free, and the RNA polymerase can bind with the promoter region and can turn on the transcription. And then the proteins will be produced: beta-galactosidase, permease, and trans-acetylase. Beta-galactosidase will convert these lactose into galactose and glucose, and then the glucose can be used happily by E. coli cell, and they can do their daily activities very easily. I hope you understood the lecture. If you have any questions, you can ask me in the comment section. I'll be happy to give you a suitable reply. Thank you all of you for listening. I wish you a happy learning. Everyone study nicely and make your dream come true. And I'll end my lecture with a quote: fight for your dreams and your dreams will fight for you. I wish you all the best, students. Best of luck to you all.